Howdy, I'm John Kaltenbach, and I'm going to talk about Arthropod Biocontrol in Colorado, including two current programs, Japanese beetle and emerald ash borer. Now in its 77th year, the Palisade insectary was founded on arthropod biological control. In 1945, the peach moth, Grapholita molesta, threatened the burgeoning peach industry in Palisade. Using an imported wasp, Macrocentris ancillaris, control was achieved and is a project that continues to this day. Building on the success of the MAC program, the Palisade insectary has continued to provide biological control solutions for Colorado and other western states. Over the years, we have worked on over 34 pests, including 21 noxious weeds and 13 different arthropods, importing and or working with 89 different agents to target those pests for control. Of the 89 agents deployed for control, beetles are the most numerous, almost 40% of the agents, a number that includes 17 weevils and 13 leaf beetles. Wasps make up more than a quarter of the agents at 28%. Two highly impactful pests that have arrived recently are beetles. The Japanese beetle, Popillia japonica, a scarab, and emerald ash borer, Agrylus planipennis, a flat-headed wood-boring beetle. The main goal of biological control is to reduce pest populations below a threshold of damage or economic injury. For both of these arthropod pests, reducing the overall populations will lower their impact where they are established and slow their spread to uninfested areas. An agricultural and horticultural pest of high impact, the Japanese beetle, referred to as JB, is a difficult pest to control. The larval form of the insect, called grubs, clips and then eats the roots of grasses, especially well-watered turf grass. The adult form of the insect feeds on over 300 species of host plants, consuming the foliage and flowers of plants, trees, and shrubs. After arriving in the early 2000s in and around golf courses and parks in the South Denver metro area, the population of this non-native pest has been growing along the Front Range. It is now well established and expanding in Colorado, with high population areas shown in red, moderate in yellow, and low numbers in the green areas. No Japanese beetles have been detected in the areas depicted in blue. There are a number of biological control agents available for Japanese beetle. Both milky spore and nematodes are commercially available. However, they have not been shown to be effective in Colorado. We are currently working with these three agents: Ovovesicula papiliae, a microsporidium pathogen, and two parasitoids, Typhia vernalis, the spring typhia wasp, and Ishtocheta aldrichi, the winsome fly. Microsporidia are spore-forming unicellular parasites and are a sister group of fungi. In JB, Ovovesicula papiliae reduces the amount of offspring produced by female beetles and also inhibits the ability of larvae to survive the winter. Ovovesicula infection can be diagnosed by the presence of spores in the Malpighian tubules, which are excretory organs that lie in the abdominal cavity of Japanese beetle. The introduction of Ovovesicula began in 2015 with Dr. Whitney Cranshaw of Colorado State University importing dead beetles collected in areas of Michigan where the pathogen was present. This was the first time dead beetles were used in this manner. The dead beetles were applied to turfgrass in Boulder and Pueblo, where Japanese beetle populations were very high. Whole adult beetles were cast on the turf. In other spots they were buried, and in others beetles were ground up to make a slurry that was poured on the grass. The three introductions in 2015 were made at Flatirons Golf Course in Boulder, and two releases in Pueblo at the Zoo and Elmwood Golf Course. In 2017, beetles were collected from those three release locations, and infection with Ovovesicula was confirmed at Flatirons Golf Course and at the Pueblo Zoo. Based on the successful results of the 2015 introductions, in 2018 more releases were made, 7 additional locations along the Front Range, and then in 2021, collection and redistribution was started from populations in the Flatirons Golf Course of Boulder to other parks in Boulder, Denver, Fort Collins, and Pueblo. In total, 20 releases have been made in Colorado. In addition to the initial 2 locations that were positive, Flatirons Golf Course and the Pueblo Zoo, Ovovesicula has been detected at Mamie Eisenhower and Cheeseman Parks in Denver and Stern Park and Hudson Gardens in Littleton. We are waiting on results of detection sampling done in 2022. The technique of introducing dead infected beetles works, as Dr. Cranshaw has successfully introduced Ovovesicula to Colorado. Research from Michigan demonstrated high larval mortality in infected populations, making this a very promising biological control. At this time, Ovovesicula may be the most feasible to collect and redistribute in Colorado, although its effectiveness has yet to be measured here. The next agent I'd like to talk about is Typhia vernalis, a parasitoid wasp. In 2018, both the CDA and Dr. Cranshaw began to import Typhia vernalis, also known as the spring Typhia, from populations in North Carolina and Kentucky. The spring Typhia parasitoid has been found to reduce populations of Japanese beetle in some states, with a parasitism rate of over 60% recorded. The female is about a half an inch in length and digs into the soil looking for Japanese beetle grubs to parasitize. Grubs are paralyzed by the female wasps, and an egg is laid on the sternum of the beetle. The wasp larva consumes and kills the grub. A total of 959 agents have been released in Colorado. We have not yet had recovery, but will continue to do surveys in the spring of 2023. One of the things we'd like to promote is the idea of farmscaping for wasps. Parasitoid wasps are more effective if the adults have plentiful, accessible food. One way we can do that is to plant species that have nectar and or can be sources of aphid honeydew, some of which are listed here. The third agent I would like to talk about is Ishtokita aldrichi, the winsome fly, another parasitoid we've introduced into Colorado. Female winsome flies lay their eggs on the pronotum of a Japanese beetle. The fly larvae hatch and burrow into the beetles and kill the beetle within 5 days. To collect the flies, we collect the beetles that have eggs on them. CDA has utilized a technique where the parasitized beetles are protected over the winter from hyperparasitization by other wasps, and then buried in the ground in a rodent-proof cage, as shown in this picture. This technique was introduced to us by Dr. Richard McDonald, the collector of the CDA winsome fly releases. His experience was that a large collection of puparia, winsome fly pupas surrounded by the dead husk of a Japanese beetle, were an attractive, delectable treat for mice and moles. The exclusion cage is buried for 2 years, with 80% of the flies emerging in the first summer and the remaining 20% coming out during the second summer. In total, there have been 12 releases in 9 different locations along the Front Range, from Boulder down to Pueblo. From 2018 to 2022, 1,932 winsome flies have been released in Colorado. CDA has released agents collected by Dr. McDonald in western North Carolina, where he established populations 30 years ago. Dr. Cranshaw has released winsome flies collected by collaborators in Connecticut. To date, 1 parasitized JB was spotted in 2022 at the War Memorial Rose Garden, adjacent to Stern Park, in Littleton, where the bulk of releases were made by CDA. There was also a picture showing a JB parasitized by a winsome fly that was uploaded to iNaturalist by someone in 2022, with a location about 3 miles east of Stern Park. We have learned how to survey for, collect, and redistribute 3 biological control agents for Japanese beetles. It is our hope that release sites become established nurseries from which we can redistribute agents collected locally. At this time, the most promising agent for redistribution seems to be Ovovesicula papiliae, and CDA is interested in measuring agent establishment and impact in the coming years. Our other fairly new arthropod pest in Colorado is the emerald ash borer, Agrylus planipennis, most often referred to as EAB. First identified in Boulder, Colorado in September of 2013, tree ring analysis indicated that the beetle probably arrived 5 or 6 years prior to detection. The most impactful forest pest ever introduced into North America, emerald ash borer has caused billions of dollars in damage. The beetle larvae feed under the bark in the phloem from the top down and are very difficult to detect. They attack all Fraxinus species, that is, all ash species, and can kill a tree in 1 to 3 years after you notice the symptoms of infestation. First detected in Michigan in the year 2002, Colorado was the furthest EAB had spread west until this last year when it was found in Oregon. The current known distribution of EAB in Colorado include the counties of Boulder, Broomfield, Jefferson, Adams, and Larimer. The only native Fraxinus species in Colorado is the single-leaf ash, found in the southwestern part of the state. However, we have abundant ash in our urban forests, as approximately 15% of our tree species, that is 1 in 6 trees, is an ash tree, and in some communities, up to 30% of the tree shade is provided by Fraxinus species. Urban trees are important for stormwater retention, shade, carbon sequestration, and even human health and happiness. Depicted here are a row of ash trees that have been lost to EAB in Boulder. You can just see the devastation. The City of Boulder Forestry has implemented the SLAM program, or Slow Ash Mortality. This involves treatment to save some valuable ash trees and the prompt removal of infested trees. The city has about 900 trees being treated on a rotational basis, 3 groups treated every 3 years, with emamectin benzoate as the product. It is unknown how many trees are being treated by homeowners, but anecdotally, treatment seems very common. They have also removed many ash trees, probably at about 1,000 since the EAB was first found in 2013. There are 4 wasps that have been introduced for the biological control of EAB in Colorado: Oobius agrilli, Tetrasticus planopenisi, Spathius agrilli, and Spathius gallinae. All 4 of these are stingless wasps. Here we see the difference in size of the wasps with an adult EAB for comparison. Oobius agrilli is an egg parasitoid, and it's the smallest at 1 millimeter long. 1 wasp kills 1 emerald ash borer egg. Tetrasticus planopenisi is the medium-sized wasp, shown as C in this photo, and it can produce up to 120 wasps per EAB larva killed. The largest of the wasps here, Spathius agrilli, can produce about 20 wasps per EAB larva killed. On the left we have Oobius agrilli, the egg parasitoid, with the EAB egg in the photo, so you can see the size comparison. On the right we have the endoparasitoid, Tetrasticus planopenisi, that uses its ovipositor to put eggs through the bark and into the larva of an EAB feeding underneath the bark. The larger wasps, Spathius agrilli and Spathius gallinae, can penetrate thicker bark with a larger ovipositor, and they lay their eggs in the gallery along with the larva, and their young consume an EAB larva from the outside. They're called ectoparasitoids. In 2015, CDA joined a study led by Dr. Julie Gould with the USDA APHIS PPQ Science and Technology Program. The purpose of the study was to see if we could establish parasitoids while systemic insecticides were used to save large ash trees. We wondered if the populations of parasitoids would increase quickly enough to control EAB and allow for the stoppage or reduction of chemical control. The study design was replicated in Boulder, Naperville, Illinois, and Syracuse, New York. To collect data on parasitoid establishment, we first used yellow pan traps and then deployed sentinel logs. To assess the health of ash trees, we recorded treatment or no treatment, signs and symptoms of EAB infestation, and crown class from 1 to 5, 1 being fully leafed out healthy ash and 5 being a dead tree. For emerald ash borer density and parasitism rates, we took branch samples. We had 2 plots in Boulder, a 1 kilometer radius release plot centered around the east campus of CU Boulder, and a control plot the same size centered on the southeast corner of the Flatirons Golf Course. In this map, locations of parasitoid releases are along Boulder Creek in purple, and the yellow dots represent locations of pan traps spread out across the 2 plots. The total number of parasitoids released for the Boulder IPM study are in this table. No parasitoids were released in 2018. As of 2028, Tetrasticus is the most abundant and has been found every year since 2017. Oobius agrilli was found in a yellow pan traps in 2019, but we stopped using the traps in favor of sentinel logs in 2020, so the status of Oobius is unknown. Spathius species have emerged from the sentinel logs the last 3 years, so they seem well established. Here is a map showing the canopy assessments from the summer of 2022. Of the 493 trees we began assessing in 2015, 182 have been removed. 109 are still Class 1, and 148 are still Class 2. Here's a table with the canopy assessment numbers for the whole study from 2015 to 2022. No canopy assessments were done in 2021. Only 47 untreated trees remain from the initial 158, or about 30%. Of those 47, 28 of them had a canopy assessment of 1 or a 2, which is pretty healthy. So some of these trees are surviving untreated. We will be sampling branches in March and April of 2023 to assess EAB numbers and the percentage of parasitism, to try and determine what's keeping these trees alive. Some lessons learned in this study. It takes patience, both for the parasitoids to establish and to be able to measure the results and impact. A complicating factor is that we don't control the pesticide applications, and withholding treatments is a difficult ask for valuable trees. We have found parasitoid establishment, and many untreated trees remain. We are excited to publish our results when we get everything wrapped up. A study like this would not have been possible to carry out without the cooperation of many agencies, including the City of Boulder Forestry, USDA APHIS PPQ, and CU Boulder. A big thanks to the USDA APHIS PPQ for providing money for both of the projects, Japanese beetle and emerald ash borer. Thanks to Dr. Richard McDonald of Symbiont Biocontrol for his help in reviving interest in Japanese beetle biological control, and to Dr. Cranshaw of CSU and Larry Hurd of South Suburban Parks and Rec for their support in Japanese beetle biocontrol. For emerald ash borer, thanks to Dr. Julie Gould for heading the study, and for the cooperation of CU Boulder, City of Boulder Forestry, and many CDA seasonal employees.